Welcome back, everyone. We begin always with a little prayer. In the name of the Father, the Son, and the Holy Spirit. Come, Holy Spirit, fill the hearts of your faithful, and kindle in us the fire of your divine love. In the name of Christ, our Lord. Amen. All right, we're continuing with the lectures on fundamental principles in bioethics, looking at the evidence for evolution. In other words, a mechanical and chemical explanation for the origin and development of life on Earth. Once again, to put it in context, the idea is to be faithful both to the scientific evidence for evolution, for example, the fossil record and the phylogenetics at the molecular level of the percentage homology between all the different species on Earth. And at the same time, faithful to our beliefs that God is creator and that somehow he's involved in the creation of the universe, in the creation of life, and the creation of the human here on Earth, okay? So the first half of the course is basically looking at the evidence for the process of evolution and trying to understand that process to a certain level, which we have to land into natural selection at some point, natural selection, okay? And then the second half will be looking at the biblical and magisterial evidence in a theological way, in a metaphysical way, for God's intervention in this process of life and particularly human life. So what we're calling the phylogenetics of the human species, the origin of the species as a whole. Any questions or comments from previous lectures? You should become familiar now with a number of terms when we say that adaptation and evolution occurs physiologically, structurally, biochemically, and also in behavior, percentage similarity in those patterns. But also considering what we call biodiversity, the many species, different species that exist today, and the similarities between them, not just the external morphology, the shape of the individuals. For example, when we talk about tetrapods, what are tetrapods? Tetrapods, animals that have 4 legs, 4 limbs, right? Okay. We're tetrapods too, in the sense that we have arms and legs, but we only use 2 of those 4 legs for walking, or 4 appendages for walking, all right? So we're bipedal, really the only animal today that is fully bipedal, that can stay walking indefinitely in principle, as long as we have energy. Every other animal that is a tetrapod, basically land animals, is going to use all 4 legs for walking. But it's interesting to see that these, tetraploidy, if you will, to having 4 legs is fairly universal for many land animals that spreads across many mammals, reptiles, and so forth. And so it begs the question of a common origin, right, for these tetrapods. So we can say that if life somehow began in the oceans, migrated to land, the simplest way to get around land walking instead of swimming was with 4 legs. And so there must have been a common tetrapod millions of years ago, hundreds of millions of years ago, that led to the development of all land animals that use 4 legs. Okay, so that's just as a frame. Now, today we're going to look at 3 origins. Actually, we're going to focus really on one origin. We're going to just very briefly the origin of the universe and Earth, and then the origin of life. And in following lectures, we'll look at the origin of humans itself by the fossil record, by the evidence that is here scientifically. So with regards to these origins, just briefly to state again that it is thought that the origin of the universe was about 14 billion years ago, more or less, 13.8 billion years ago, by the theory of the Big Bang, or that moment of singularity, from nothing, something, not just something, but everything, actually, from nothing, everything, okay? So ex nihilo, from nothing. And at least scientifically, there's evidence for that. Looking at the expanding universe of today, if we reverse that clock, hypothetically, we would come to a moment of contraction of, let's say, total contraction of all matter and energy in the universe back about 14 billion years ago. We'll look at this a little more detail in the environmental course, environmental bioethics, okay? Basically two phases, generally the two phases, the inflation and the expansion of the universe and so forth. But for now, just let's leave it there because it's not the focus of this lecture. Rather, the focus of this lecture is going to be the origin of life on Earth and the possible development of that life into the species that we see today. And for later, the origin of the human, at least at the level of the genus, Homo. For example, here you see 4 skulls, which are definitely Homo skulls, and yet their shapes are quite different, right? So it's pointing to perhaps 4 species of the same genus. These would be like cousins, if you will. They may be on the maternal side or the paternal side, they have the same last name, but different individual names. All right, so for the origin of Earth, we need an Earth that is cool enough to have liquid water, because when we look around, we see that liquid water is the basis of the possibility of life on Earth. Liquid water, all right? In other words, at the chemical level, basically life is metabolism, which is a series of complex chemical reactions that are occurring typically in a sequence, in an orderly fashion. And so very briefly, the formation of the Earth, which is typical of planets, is a process called accretion, all right? And accretion is, if we can go back about 4 to 5 billion years ago, there is matter that is flying around the universe, chunks of rocks. Some are small, maybe as small as our fist, some as large as this building, others as large as maybe, you know, 1/10 of the size of the Earth or something like that. So these rocks, boulders, this matter that is flying around, okay, from explosions of previous galaxies, of previous solar systems. And because gravity is one of the 4 fundamental forces, right, in the universe, this matter exerts an attraction toward other matter. And so these pieces of rocks and boulders that are flying around, when they intersect, they start colliding and conglomerating, right? They're typically very hot. They could be in a molten state. And their trajectory typically is a linear trajectory, because unless they go through a gravitational field where they start bending into that field, depending on the amount of matter that they have and the density of that matter, right? So there's that little question, what weighs more, a pound of lead or a pound of feathers? Which one weighs more, the pound of lead or the pound of feathers? Depends on what? It's a pound, right? So a pound of lead weighs the same as a pound of feathers. It's just that the pound of lead is going to occupy a small amount of space, and a pound of feathers could occupy maybe this whole room. All right? But a pound is a pound. So different density rocks and boulders, they start attracting each other if they intersect the pathway of their gravitational field. And so colliding and coalescing this molten mass is a process of accretion. And this is basically, it's considered to be pretty much the standard way how planets are formed, opaque bodies. And so the planetary system of our solar system is no exception. Very briefly, including the 4 major planets, the 4 minor planets, and the 4 dwarf planets that have been discovered so far of our planetary system, okay? So we have this accretion process going on that for the Earth and the other planets in our solar system, Venus, Jupiter, Saturn, etc., happened about 4.5 billion years ago, just under 5 billion years ago, okay? For example, the moon. The moon is one of those chunks, mass that was flying around that got trapped into the gravitational field of the Earth, but did not get trapped close enough to get sucked into the Earth. And so it's orbiting. And it is a balance between 2 forces that oppose each other. And when they oppose each other, the result of those 2 forces is a vector, right? And so the 2 forces, what are the 2 forces on the moon right now? It's a dynamic state, really, as stable as it looks. I don't know if you've seen the moon the past couple of days, a couple of nights, full moon, beautiful, right? Okay, so, which by the way is the reflection of the sun on the moon, okay? Because the moon is also an opaque body. The 2 forces that are keeping the moon orbiting around the Earth. In principle, those are 2 linear forces, but they're in opposite directions, not quite opposite directions. They're kind of tangential to each other. And so they generate a vector, which is the result of the 2 in a diagonal. The 2 forces are, the most obvious one, gravity. From where? From the Earth to the moon, right? Because the moon also exerts a gravity upon the Earth, lesser gravity. But the other force that is keeping the moon orbiting around the Earth, it's its own velocity, its speed, okay? And keep in mind, this is space, right? In other words, there's little to no friction in space. And so there's nothing to slow down the moon from wherever it exploded, okay, as it's wandering through the universe. It's essentially maintaining a constant speed, a velocity. But that constant velocity, that trajectory at some point came close enough to the gravitational field of the Earth to get caught in that gravitational field, but not too close so as to collapse into the Earth, you see? And so it's a balance. Other matter, other moons, let's say, that were flying around the Earth, they were either got too close into the gravitational field and got sucked in the process of accretion and became part of the Earth, or they were too far away from the Earth and they may have been affected by what would the gravitational field do on another object, a meteorite or an asteroid flying by, but not getting caught into the field. It may divert it may change its angle of trajectory a little bit because of the gravitational pull, but because of its own velocity, managed to escape the gravitational field. It just kind of buzzed it nearby, but did not get trapped into the field. So at most, it may have bent its trajectory a little bit and then continued again in a linear fashion, all right? And so there are 3 possibilities basically when other matter is flying around a planet that is in the process of accretion. The 3 possibilities is that matter either gets sucked into the planet or flies by and doesn't get trapped into the field, right? Manages to escape the accretion, or gets trapped into the orbit. which is what happened to the moon. It's kind of surprising that it only happened to one object so far. It could happen to another object that will come this way eventually, and we may have a second moon, for example, at a different distance, in a different orbit, and so forth. And maybe at some point those two orbits collide. So really, the universe is in a dynamic state of coalescing and exploding and so forth. It's just that because the distance is so large and time is irrevocably tied in with distance. In fact, Einstein talked about the space-time continuum, right? That we don't normally in our lifetime, we don't experience these events. Probably it's a good thing because they tend to be catastrophic events. Anyway, you see what happens there. Another interesting thing about the moon, and I'll leave it at this, you see how the moon is all full of holes, craters, right? And the moon has been around for about the same time, more or less, about 4.5 billion years. And so it's interesting, the moon is informative in the sense that what we see on the moon is what should have happened to us. In other words, those craters are mostly bombardment from other asteroids that were flying around, smaller than the moon, that got sucked into the moon and bombarded it. And it's all full of craters. Those craters are large enough miles distance in diameter that we can literally see them with our naked eye from the Earth, okay? So those are huge craters, and invariably at the center of every one of those craters, deep down, maybe 1, 2, 5 miles down in, is going to be the meteorite or the asteroid that hit, and it's embedded in there, okay? The Earth should look the same, all full of craters around it, and it doesn't. And we're getting bombarded essentially with the same kind of matter that is hitting the moon, right? So the moon is like an experimental place. Why don't we look like the moon? And instead we have this beautiful living Earth full of life because of a very thin layer that is around the Earth, that is a gas, and it's called the atmosphere. The atmosphere is only, well, the troposphere, the closest atmosphere to us is about 10 kilometers in height. And then it gets thinner and other gases there. But basically those 10 kilometers is a filter. It's a very effective filter. It burns because these asteroids and meteorites are coming at such speed that when they enter the atmosphere, specifically with its oxygen content, they burn up and they explode into 1,000 little pieces. And some of those pieces actually make it through the atmosphere and hit the Earth, and those are the meteorites and the asteroids that are found periodically, you know, a chunk that goes either, typically they go into the ocean because 3/4 of the surface of the Earth is ocean, okay? That's another good thing because those meteorites come at an incredible fast speed and heat. But you get the picture, right? That there's a dynamic state, but it's long-term. And so everything seems to be very static and stable, and every night the moon is there and so forth, but really it's pretty dramatic. And it's estimated that in a few million or hundreds of millions of years, the moon will probably collapse into the Earth. And that will likely be the end of life on Earth, as we know it anyway, because it's a pretty significant a chunk of mass that is out there, okay? All right. So the process of accretion, bottom line, about 4.5 billion years ago, it was a red hot magma, and in some, at the core, even white hot. Again, in the environmental course, we'll look at it in more detail, the layers of the Earth down to the crust, down to the core. But we're concerned with the crust. The crust, in other words, the outer layer, think of the peel of the orange, right? That had to get cool enough to hold liquid water. In other words, it had to cool down to the range of 0 to 100 degrees centigrade, which is a very mild temperature when we compare the drastic temperatures, the extreme cold and heat that exists in the universe, all right? And that happened about, it took about 500 million years, more or less, less than 500 million years for the Earth to cool down enough, radiating heat, right? So that we could have a crust that at the surface of the crust, what we call the continents today, was cool enough to hold liquid water. And so that was about 4.2, about 4 billion years ago, more or less, 4 billion years, all right? And it seems that quite soon after that, life began because we have fossils that go back that time, all right? So I want to introduce you to zircon. Zircon is interesting because this zircon crystals, here's a zircon crystal, all right? It contains carbon, but it also contains uranium. And it's exciting because carbon is thought to have come from organic life. For example, graphite, which is what makes the lead of the pencil, right? There's no actual lead in there. Typically, it's graphite, it's carbon. There may be some lead, I really don't know. But it's mostly graphite, which is carbon, essentially fossilized carbon from organic material. Remember, organic matter needs the element carbon, right? But the other interesting thing, because it has uranium, because the zircon has uranium, the half-life of uranium, of U-238, 238, is about 4.5 billion years. So it's a very handy element for measuring the date, for dating this graphite and these zircon crystals that contain carbon that is thought to be organic carbon. In other words, carbon that was used by organisms for the formation of life, okay? And it comes to about the zircon, zircon crystals come to about 3.8 to 4.4 billion years ago. Then what happens? Well, scientists have actually found fossils that go back to about 3.8 to 4 billion years ago. Okay, they're called stromatolites, and they're layers, these layers. They looked at sediment layers, and they're sediment layer, but it turns out that on top of the sediment, right, which is mineral, sand, and silt, on top of that layer, this yellow and black, all right, is actually organic material. There are organic compounds, and it is thought that these organic compounds are decomposed archaea. The archaea, which are bacteria-like organisms, but more primitive than the bacteria. And so, for example, these stromatolites are found in Australia in a bay that is called Shark Bay, right? There's an actual photo of them. These boulders are sedimentary rocks that have these layers. Aside from the mineral, sandy, silty layer, which is the lighter black and white here, the mustard color and the black color are organic compounds that are thought to come from these archaea. And the archaea look very much like bacteria, have very similar structures. They have a cell wall, they have a cell membrane, they have a cytoplasm, they have DNA. Okay. The DNA is not enclosed in the nucleus, so they are prokaryote. Excuse me, they are prokaryotes. A little bit of Greek here. Karyo, the word karyo means nucleus in Greek. And pro is before nucleus. So even though there's a concentration of DNA in some area of the bacterium or the archaea, there's no nuclear membrane, okay? Now, they look very similar, so why are they different? And why are archaea archaic, in other words, older than bacteria? Well, it turns out that on the cell wall, the archaea are missing a middle layer. The cell wall of bacteria is a 3-layer wall. There's a lipid layer on the outside, and there's a plasma membrane on the inside. And there is this middle layer that is called peptidoglycan. Peptidoglycan is a complex organic molecule that is made up of peptides and carbohydrates, so organic molecules, right? But the archaea are missing that middle layer. And that's why it is thought that the archaea evolved before the bacteria, because the bacteria have an extra layer, okay? So this extra layer developed at some point, and archaea became bacteria. But some archaea still are living today and are found. Typically, they are extremophiles, all right? And extremophiles are, now we go to Latin. Philos or philia is love, and extremo, they love extremes, okay? So these, essentially these archaea like to live on the dangerous side of life, on the edge. For example, there are halophiles. Halo is a reference to salt. They live in salt marshes where the density of salt is very high. For example, the average salt density of the ocean is about 35 parts per thousand of total salts, right? 35 parts per thousand of salts. The Dead Sea in Israel is dead because it has no life except for halophiles, extremophile bacteria and archaea that live there. Dead Sea is very interesting because it's disappearing. It's divided into two parts now, and it's the bottom of where the Jordan empties, the Jordan River empties. Here's the Sea of Galilee, which is now freshwater. It's also the Lake of Gennesaret, where Jesus began his ministry. Anyway, there's a lot to say about the Sea of Galilee, but basically the Jordan River goes north-south, okay, and trickles into the Dead Sea, and has been doing that for millions of years. And so the Dead Sea has a very high salinity, about 10 times more salty than the ocean, than the average ocean. And so one can actually float in the Dead Sea. You see these chunks. Looking for a picture. Yes, on the Dead Sea, you see these chunks of white rocks floating around. And you think that at first sight, you think that it's like snow or chunks of ice, little tiny icebergs that are floating around. However, it's very hot there, okay? And so it's certainly not ice. They're chunks of salt that are floating around. And it is so salty that people can actually, you can float standing up. This guy is horizontal, but if you turn vertical, you can float standing up. It's so salty, it's dense, it's like, it's viscous, like baby oil. And you can see it's very clear, crystal clear, because basically nothing lives there except these extremophiles, these halophiles that love salt. Anyway, it's an interesting phenomenon. It's also about 200 feet below sea level, and there's a project to fill it with salt water and so forth. It's controversial. But, just to point out that today we have examples of these extremophiles, all right? In other words, archaea and bacteria that could live in extreme conditions, conditions that would be similar to the crust of the earth 4 billion years ago. which were very different conditions from the conditions of today. It would be much hotter and high concentrations of salt, for example, because of all the minerals that are spewing out from volcanic activity. And the atmosphere was certainly very different. A lot of ammonia, methane, CO2, no significant amount of oxygen around, okay? And water vapor and hydrogen, which is very explosive and reactive. There are also other extremophiles that are thermophiles that tolerate extreme temperatures, like for example in geysers, geysers or geysers, how do you say that, geyser? For example, out at Yellowstone National Park, all right, Old Faithful, the geyser that's still spilling out after thousands of years. It's essentially water that percolates down, touches the mantle, touches the molten rock down there, vaporizes, and then comes out as water vapor, finds a crevice to come out as a geyser, and it does that periodically in a sequence, in a cycle. And so there are extremophiles that live in very high temperature. They're called thermophiles. There are also extremophiles that live in a desertic area, essentially no water, no significant amount of water available. They're called xerophiles, okay? Reference to the desert. There are also radiophiles, in other words, bacteria and archaea that are resistant to radioactivity, to UV, right? And some are what are known as polyextremophiles. There are a couple of archaea that have been discovered on Earth that actually have all of these characteristics. They tolerate high heat, high dryness, high salinity, high UV. What's interesting about these polyextremophiles? Let's say that, and these have been found, I had an example of it. Anyway, what if we take some of these polyextremophiles in a vial and we send them to Mars? They could begin a process that is known as terraforming. You've heard of terraforming planets. Terraforming planets is to experiment with other planets, which today, as far as we can tell, don't have any life, organic life as we know it, and seed that planet with some polyextremophiles to see if they take off in the evolutionary process, and millions of years from now, we get some kind of recognizable organic life, similar to the life that we have on Earth. Terraforming, it's an exciting science, it's controversial, and it's bioethical. Do we have a right to terraform another planet? And who knows what will come out of there and what alien we may be creating, right? So it can quickly go into big sci-fi, but I can tell you that there's a scientific basis to it because of these polyextremophiles. And they're being studied under labs that are P4 labs, right? Like the Wuhan lab, that nothing is supposed to get in and out of that lab, all right? P4 is a high security lab that is the highest level of security, okay? Where nothing is supposed to get in and out of that lab. Anyway, enter cyanobacteria. Cyanobacteria have been dated back to about 3.8, 3.9 billion years ago, all right? So what's interesting about cyanobacteria is that they photosynthesize. They are bacteria, so they're prokaryotes. They're relatively simple organisms. They could have derived from archaea, but they have this extra characteristic that they photosynthesize. They have these thylakoids, which are photosynthetic pigments, and therefore they metabolize CO2 into oxygen and organic molecules, all right? So they take CO2 and water and do the photosynthetic alchemical reaction to produce oxygen and organic material, typically glucose. So what is interesting is that it's considered that the current atmosphere that has a relatively high percentage of oxygen, about 16% oxygen, all right, was produced by the cyanobacteria about 3.8 billion years ago. It's amazing. And because the primitive atmosphere had a relatively large amount of CO2, carbon dioxide, precisely from the ash and the volcanic activity that was occurring all over the earth as the crust was beginning to cool down. Okay? So imagine the amount of cyanobacteria that was necessary to produce the oxygen that we have in today's atmosphere, which makes it a benevolent atmosphere. But this basically sets the possibility for the explosion of life in the diversity that we see today. So the two basic ingredients, liquid water, and then oxygen gas, O2, diatomic oxygen, available for respiration, for aerobic respiration, which is the counter reaction of photosynthesis. Between the two, the complementary reactions, you know, they're called redox reaction, okay? So photosynthesis takes water and CO2, turns it into glucose, and the byproduct is oxygen. And then cellular respiration, what our cells are doing right now, and the cell of every living organism, including plants, their plants are alive, so they also respire, right? Metabolic respiration takes the glucose, takes the organic molecule with oxygen, burns the glucose, and the product is CO2, carbon dioxide, and water vapor that is released into the atmosphere. And in that burning of the glucose, energy is released, which recharges the ATPs, the triphosphates that allow for metabolism in the body. So there's a mechanism that occurs as a biochemical mechanism. It's elegant, and it affords the possibility of life, metabolism at the organic level. All right, so cyanobacteria come into the picture, tons and tons of cyanobacteria covering the earth in a liquid environment to produce the oxygen that we have today. So they transform the atmosphere into a benevolent, from a toxic atmosphere to a benevolent atmosphere, amenable for life. Another step that occurs in the process is compartmentalization. It's a big word. Compartmentalization, meaning membranes within membranes. In other words, specialized work, right? You notice that a car, if we look at a car, inside a car, things are not haphazard. Inside the chassis of a car, things are in place in order for the car to work. There's order. So the motor typically has the pistons, or if it's an electric motor, it will have the rotor and so forth. It's in a particular place. And then that motor is going to be attached by some mechanism to the tires that are the ones that turn. Then in another place of the car, there will be a cabin for the driver to drive the car and so forth. So there are compartments, right? And there are compartments all over the place because we mimic nature basically in our artifacts and the way we do things, even in a house, in a home. Typically in a home that is organized, you know, you don't have just one common living space where people eat and go to the bathroom and sleep all in one big place like they used to back in the cavern play in the primitive living. No, there are compartments, and the living space, the cooking space is different from the sleeping space and so forth, right? So these compartments are also the way for life to occur within the cell, so that different chemical reactions can be kept one from the other in an orderly fashion. And we can have essentially self-functioning or metabolism. That requires membranes. membranes within membranes to form these compartments, to form these rooms, all right? And we have one bacterium today, for example, this Gemata oscuriglobus has these membranes, as you can see here, there's a cell membrane and a cell wall, external, and then it folds in. You can see that it folds in here, okay? Comes out again. There's another fold in here and makes these compartments, makes these cells within a cell, all right? These compartments can then have different activity going on from each other because they are in a discrete area of the cell. And so this process of membranes within membranes is a process that is known as endosymbiosis. Okay? It is thought that this process of endosymbiosis is what gave rise to at least 3 organelles that we have inside our cells. Now, just like our body has organs and our organs keep us alive and different organs have different functions, right? The heart is a pump, the stomach is a digestive process, the neurons, the brain is for thinking and so forth. Different organs have different functions. The same way in the cell, different organelles have different functions, okay? And so they're tiny, it's a way of looking at organelles, is tiny little organs inside the cell that have different functions. And so the 3 main organelles in the cell, whether it's a plant cell or an animal cell, that have very specific functions critically associated with life itself, with maintaining metabolism, are the nucleus, the mitochondria in plants and animals, and then in addition, another organelle only in plants, that is the chloroplast, which is the photosynthetic pigment. So here is a representation of an idealized animal cell and plant cell, 2 or 3 different differences between them. First, the commonalities. Both plant and animal cells, they have a nucleus that is it's an enclosed DNA compartment, all right, with a membrane, a nuclear membrane that is a double membrane, just like the cell membrane is a bilayer. It's a bilayer. Interesting. Why would the nucleus be a bilayer? A single layer would be sufficient to contain the DNA, but no, it's a bilayer. And it's a bilayer that has essentially the same composition as the membrane of the cell itself, as a cell membrane. Okay, it's a phospholipid bilayer, right? For plants and animals. So it's, the thinking is that the nucleus evolved before plants and animals differentiated, okay? Because it's a common characteristic between both. The same argument can be made for the mitochondria. We have several thousand mitochondria in each cell of our body. Okay? Mitochondria, one mitochondrion, several mitochondria, they have also a bilayer. But in contrast with the nuclear bilayer, which is flat together, the 2 layers together in a spherical shape, more or less, the bilayer of the mitochondrion is a little different because the outer layer is smooth, but the inner layer is corrugated, all right? So maybe this is a little better. See, the outer layer is a smooth outer layer, but the inner layer is corrugated. It folds in and out. What does that do? That increases the surface area on the inside, which is literally where the chemical activity occurs of respiration, of burning the glucose to produce the energy for recharging the ATPs, the adenosine triphosphate. So again, it's a bilayer organelle that is thought that was formed by this endosymbiotic process. Similar argument for the chloroplast. only that for the chloroplast, the structure is even more complex. It has little discs inside, the thylakoid discs, that contain the actual photosynthetic pigment, the chlorophylls, the alpha chlorophyll, the beta chlorophyll, and so forth. And that's where the photosynthesis occurs, which is trapping light energy and turning it into chemical energy in the bond and the carbon-carbon bond of the glucose. So it's an extremely elegant process. It's amazing. The level of order in a single chloroplast is just bewildering. How this little tiny super microscopic organelle manages to trap light energy and transform it into chemical energy, potential energy, right? That is available then for work in the cell. And that work, again, we call metabolism. And another word for metabolism is life, organic life as we see it, all right? But the chloroplasts only occur in plants. And so, again, it's thought that the chloroplasts develop later, possibly an endosymbiosis of cyanobacteria that got trapped into some animal-like cell that now had the capacity to photosynthesize also. Because you notice that plant cells, plants are also alive, right? So they also need to respire. They also need to burn glucose to get energy to live. And they have mitochondria to do that. Okay, but they produce an excess of glucose, of course. So that's why they're considered producers in nature, as opposed to, in contrast to consumers of food, which are the animals. Just for the sake of completion then, another difference, a couple of other differences between plant and animal cells is that plants also have a cell wall in addition to a cell membrane. Now the cell membrane is soft. You can consider like a balloon filled with water. It's soft, but the cell wall is rigid because it's mostly cellulose, long fibers of cellulose, and it's like taking that water balloon and put it inside a shoebox, more or less, okay? So the cell wall is rigid, and that allows plants to be much larger than animals, like trees that can be 2, 3, 4, 10 stories high, much, much larger than animals. Because the cell wall is rigid and literally it's one stack on top of the other, but it also makes them more rigid. And so typically plants are sessile, they're not motile, they're stuck on the ground, they don't move around, but animals have motility in part because we don't have cell walls, all right? And so the membrane of the trillions of cells that we have is flexible enough to allow movement. And just finally, also, plant cells typically have this blob inside that is called a vacuole, typically water or some kind of fluid, right? A vacuole is like a storage space filled with fluid, typically water, that animals don't have. Okay, let's move forward. Let's take a look at this little video here for endosymbiosis. I may have it already downloaded here. Endosymbiosis, let's try to do this. Go here. Oh. Okay, now what I need to do is I need to go to optimize video, which may minimize my speaking or my visual, but this is for the sake of the video, okay? So Chris, hang in there. Hopefully you'll be able to see the video okay. Sounds.
And some of these when we look inside of eukaryotic cells, we see membrane-bound organelles. And some of these membrane-bound organelles are particularly interesting. For example, here is a diagram of a chloroplast. They're found in plant or algal cells. And we know that this is where the photosynthesis takes place. But what's really interesting, above and beyond that, is that it seems that chloroplasts have a lot of the machinery necessary for being a prokaryotic cell on its own. We don't see it acting on its own, but it has its own DNA. It has ribosomes, which we know are the site where we go from messenger RNA to protein. Similarly, another interesting membrane-bound organelle that we see in eukaryotic cells, and this would include even animal cells and the cells in your and my bodies, are mitochondria. And mitochondria are often viewed as the energy factories of eukaryotic cells, where we can leverage oxygen in order to produce ATP. And like chloroplasts, mitochondria has its own DNA. It also has mitochondrial ribosomes. Here are some just diagrams of how mitochondria might look inside of a larger eukaryotic cell. And so evolutionary biologists for many decades looked at this and said, well, why do these things exist? Why do they almost look like prokaryotic cells on their own? And there's even examples of prokaryotic cells, independent prokaryotic bacteria that live in symbiosis inside of other cells, and they look an awful lot like mitochondria and chloroplasts. And so if we fast forward to the 1960s, someone named Lynn Margulis comes on the scene with Endosymbiosis Theory. And her view is that these membrane-bound organelles like mitochondria and chloroplasts, if we go deep into our evolutionary past, say 2.5 billion years ago, their ancestors were actually independent prokaryotic organisms that could produce energy aerobically or using oxygen. And precursors to what we would consider today to be modern eukaryotic cells that might have already had some membrane-bound structures like a nucleus and maybe some other things, that they could only metabolize things anaerobically. They couldn't leverage oxygen, while these other characters could leverage oxygen, and then they could have become symbionts, where the one that could leverage oxygen to produce more energy would get engulfed into the larger cell, and that larger cell is able to provide nutrients and protection, while the smaller cell that's engulfed inside of it is able to better metabolize the nutrients and leverage oxygen to produce more energy. And that over time, this symbiotic relationship became even more connected so that the smaller organism could not operate by itself, that it lost some of its DNA that was necessary to act independently, and some of it might have gotten incorporated into the DNA of the larger cell. And those smaller organisms are what eventually evolved into what we consider today to be mitochondria. This is a fascinating theory, and it's actually been proven out. When Lynn Margulis first published this in the late 1960s, she wasn't taken that seriously. But in the decades since, it's been validated as we've looked at the DNA structures of mitochondria and chloroplasts that this actually is the most likely theory of how they emerged in our cells. And so it's just a fascinating glimpse of evolution in general. We talk a lot about natural selection and the role of variation and mutations, but Lynn Margulis introduces another idea that could catalyze evolution, and that's that of symbiosis. And we see symbiosis throughout the natural world. And her argument is sometimes those symbionts can become so codependent on each other that they merge into one organism.
Okay, interesting, the theory of how we have acquired, excuse me, at least 3 of the main organelles, and it has to do with this fact that they have this double layer. The membrane is a bilayer by the process of symbiosis, because necessarily they would have to be engulfed. They would go through and being engulfed by the membrane of the host cell. And that would add that second layer, right, on the outside for the nucleus, for mitochondria, and for the chloroplast. So these are possible events that have an organic explanation. What I'm getting at here is that slowly over time, these processes could have produced more and more complex cells. all right? And so we go from single cells, first from prokaryotes, now to eukaryotes. Notice the word, again, karyo meaning nucleus. Eu, the prefix eu, means true or real. So real to nucleated cells meaning that they have a cell membrane, right? It's the next level of complexity. Compartmentalization is occurring. Other membranes that migrate into the cell are what is known as the endoplasmic reticulum, or the ER, endoplasmic reticulum, which there's smooth endoplasmic reticulum, rough endoplasmic reticulum. Basically, that's the site where ribosomes are lodged. And the ribosomes, like the little video said, are the machinery for making proteins. From the messenger RNA, the machinery for making proteins. So again, there's an organic process that is occurring here that has a logical explanation. Moving forward, the next step in the complexity of organisms is to form multicellularity, all right? So for example, we can begin with single cell creatures known as the protists. Remember that I mentioned that protist is a grab bag on the 5 kingdom scheme, right? That we have plants, animals, fungi. Then we have this bag of protists, and then we have the bacteria and the archaea together, known as the Monera kingdom. But here, the protists are typically unicellular. And it's a grab bag because now through phylogenetics and molecular phylogenetics, it has been found that they belong to different classes and orders and even phyla all over the place. So they're not really related. The only common characteristics that the protists have, or the main common characteristic, is simply that they're unicellular. So they're microscopic. And these are the familiar creatures that we've been hearing since high school: the amoeba, the paramecium, the euglena. The euglena is interesting because the euglena is motile, moves around, even has a long flagellum, a tail-like structure, certainly motile, but it also has chloroplast. So it moves and it has chloroplast. So it's kind of a hybrid between plant and animal, but it's neither really. Then there are also the plasmodium, for example, Plasmodium vivax, the one that causes malaria, that parasitizes RBCs, red blood cells. Radiolarians, Giardia, that has 2 nuclei, Foraminiferans. Many of these are actually plankton in the ocean. The upper foot or half a foot of the ocean surface is full of all these microorganisms. Some are animal-like, some are, which would be zooplankton, some are plant-like, photosynthesizing, they are phytoplankton, all right? But many different varieties. And some of these actually become colonial, meaning that some of these single cells start clustering and forming basic colonies. One very conspicuous one is the algae, the different groups of algae, whether it's brown algae like sargassum, the seaweed that we find along the shore here in South Florida, or rhodophytes. These are red algae. Chlorophytes, they are green algae, and so forth. And the algae, because these cells colonize, then they eventually become large enough that we can see them by the naked eye. But they start as microscopic organisms that are clustering together to form these colonies. Also, some primitive organisms that form, that are multicellular are the Cylindrates or Cnidarians. These are all of the anemones and jellyfish and polyps and corals, whether it's soft coral or hard coral. All of them are classified into this phylum known as the Cylindrates. the Cylindrate from the reference of coelom, which is a body cavity, right? It's the beginning of a digestive system, but a single opening. So for the Cylindrates, the single opening means that the mouth and the anus are the same opening. And then they have a bag, basically a compartment for digestion. When you look at the Portuguese man of war, because it looks a little bit like there is, have you ever gotten stung by a Portuguese man of war? It's bad news, okay? They have all of these Cylindrates, they have cells, thinking cells, that is a neurotoxin. They basically paralyze their prey and keep it alive, but paralyzed. And then they, with the tentacles, they suck up that prey. The tentacles are, they coil and uncoil. So the tentacles come up to this mass here at the base of the float. And here they are cells that are digestive cells. They pour out enzymes on the prey, and the prey gets digested away, and then the nutrients get absorbed into the animal itself. So it's pretty voracious, pretty nasty. The float is full of mostly carbon dioxide and carbon monoxide, which is a byproduct of the respiration that's occurring in the animal. But this is actually a colony of cells. This organism is actually, it's like a condominium, all right? It's a colony of cells that are specialized. So we see cellular specialization at the colonial level, right? And they're pretty primitive. The reason why they call Portuguese man of war, because early on, this float actually sails. It has a sail-like structure on it, and it sails with the wind on the surface of the ocean. But I want the ship. So that's the actual Portuguese man of war. It was a war. It's a battleship from Portuguese colonial times, which was just riddled with cannons. It may have actually 2 layers of cannons, and it was pretty deadly in its own time. It was a warship, and so because it was so deadly and so vicious, then this guy is called the Portuguese man of war. The tentacles, you can see the float 10 or 15 feet away on the shore or on the surface. You say, oh, that guy is far away from me. Fine, but don't be downwind of it, because the tentacles can be 10 to 15 feet long. And you see the float over there, but if you are down current of the float, the tentacles could be close to you, all right? So you gotta watch out for them. On the other side, if you wanna live on the dangerous side, and they wash up on shore sometimes, right? And you just see the little float and then the mass underneath. If it's wet, the mass is very, very stingy. Don't touch the mass, but you can grab the float. The float doesn't sting. You wanna play with it, but don't play with it. Just to finish the story, turtles, sea turtles, are, they love this inner mass of the Portuguese man of war, sea turtles, okay? And so when they find a Portuguese man of war, they go for it and they eat it, and they get all stung and their eyes bulge out like this, you know, but they love the stuff. So it's like eating hot pepper or tacos with very hot sauce, right? You get stung, but you can't stay away from it. Anyway, those are marine biology stories of nature and how exciting it is. But my point here is that we can go to multicellularity from protists, from single cell, as these cells start colonizing, forming colonies. And then what happens in the colonies, different cells specialize in different activities. So the tentacles are hunting, and the mass underneath is for digestion. It's like an external stomach. And then the float on top is for sailing, for motility. So there are different cells or clusters of cells that specialize. It's the beginning of specialization in multicellularity. Fast forward all that program through evolution, And you can get from primitive plants like bryozoans. Bryozoans are mosses, right? All the way to flowering plants, which are the most complex plants on Earth. The flower being the reproductive structure of the plant itself. In the other kingdom of the three eukaryotic kingdoms of the plants, fungi, and animals, you can start with single cell fungi, which are yeast, the one that brewer's yeast, for example, that leavens the bread, right? They form colonies, they cluster, and eventually in that clustering through the process of specialization, cellular specialization, you can come up with some pretty darn big fungi, which is mostly proteins. They're hyphae, they're cells that are long filaments, and looks like a plant, but it's not a plant because they don't photosynthesize. But they're not animals either. They're a third kingdom that is called the fungi, and they are saprophytes, meaning that they feed on dead organic material, and it's a good thing because they, as the forest dies and decays, and as animals die and decay, a big part of that decaying process is the saprophytes, bacteria and fungi that eat up the organic material, digest it down to nutrients, and those nutrients then are used in part by the fungi and in part by the other plants who are still living for their own livelihood. We can also go from primitive organisms, like I mentioned, the cnidarians, the Cylindrates, that are essentially colonial, to the largest mammals that we have on Earth, pretty complicated metabolism that goes on with actual organ systems, the compartment of compartments, from cells to tissues to organs to actual organ systems. Okay. So there's a logical progressive explanation for how we can go from very primitive archaea to bacteria, from archaean bacteria, which are prokaryotes, to eukaryotes. Well, first photosynthesis, again at the prokaryote level. And then through the process of making compartments inside the cell, membranes within membranes, we can get to eukaryotes. And then from eukaryotes, the next big division was between plants and animal cells. And somehow fungi also fit in there. Unicellular to colonial, from colonial to multicellular, and from multicellular eventually to the complexity of life that we see and the variety and diversity of life that we see on Earth today. So we can make these cladograms of the nodes are common ancestors, the nearest common ancestors, right? And we can do this by comparing the molecular phylogenetics, the percentage homology, either on the protein sequence or at the amino acid sequence of the protein, or its background to that is going to be the triplet code of the DNA. And the top surface here is the present, with the more or less 2 million species variety that we have discovered, clustered into its phylogenetic groups, and then common ancestry determined by percentage homology. Down, we can build this cladogram. All the way, we take it to the very origin, speaking of a common root, a common single organism that gave origin to all of the living organisms today in about 4 billion years. We also know from the fossil record that only about half a billion years ago, there was a huge explosion of diversity, because we find from one layer to the other of the strata that very few species in this layer of the sediment, and then just a couple million years later, many more species, fossils, are found in diversity. and this layer. And that marks the difference between the Precambrian and the Cambrian era, which is estimated to be about 540 million years ago, just a little over 500 million years ago, okay? Which is fairly recent because you think of the 4 billion, 500 million, there was one of these expansive, what are known as expansive radiations, okay? Or you hear the term the Cambrian explosion. There have been other explosions like that, but not as dramatic. This is the most dramatic just from the fossil record that is found. And so when we start looking at those fossils, we can come up with cladograms that go into older times. And the rise of the metazoans here, the rise of the metazoans, about 850, this is million years ago. And then the Cambrian explosion here. So you can see how the expansive radiation occurs in this region from 635 to 545 million years ago, the transition into the Cambrian. And all the branching, and then the branches that don't continue to the present, this is the present here, with about a dozen major phyla here. The branches that don't make it to the present, these are extinct, all right? And Meyer makes this point that every current species, its fate eventually is to become extinct. But in the meantime, the possibility that these current species may give rise to new species by the process of variation and selection on the best adapted, and that's what we call speciation. So again, it's a dynamic process that is occurring, but it is slow and it takes what is needed is some selective pressure to come into the system, whereby the most fit is the one that is most likely to survive. So we'll talk about fitness later on, but basically just keep it there in the back of your mind that it is fitness that allows an animal to adapt, especially when there is climatological change. If the environment stays pretty much the same, if the climate stays pretty much the same, the current species of that time stay about the same. But when there is climatological change, then there are two options. The animals or the living organisms either adapt or don't adapt. If they adapt, they could lead to speciation, to new, better species, more fit for that new climate. Or if they don't adapt, then extinction is the pattern. Okay, let me see. Now, yes, I have a series of videos now that I want to show you because I want to look a little bit in detail at the back side of the handout for today, which is 7 steps that offer for the possibility of the beginning of life, of making a cell, all right, a living organism, a living cell, starting from inorganic matter, starting just from the basic elements that we found that are found in nature, from inorganic material, inorganic matter, okay, elements, coming together in 7 orderly steps. The end result would be essentially a living cell. All right, so the second part of the lecture is going to be devoted to this mostly, with a few videos that I have. So maybe this is a good time to stop and let's review here the front of the handout. I talked about the origins, focusing on the origin of life itself. And then just a review here of the organic molecules. Did I talk about the organic molecules already, the macromolecules? Right, the 4 basic groups. Yeah, okay. So this is, when we get to these organic molecules, these are the necessary ingredients for having metabolism, for having order, this order, which is also, it's a little philosophical because you consider the first law of thermodynamics, which is increasing entropy or chaos, right, or disorder rather, in the universe, which is, for example, dissipation of heat. The heat that the sun radiates out, not the light, but the heat, and the heat that we radiate out also as the planets continue to cool is lost, is dissipated, radiates out, okay, so from a core in all different directions, 360 degrees around the sphere, radiates out into the universe and dissipates, it's lost. So that heat, which is a type of energy, is unavailable for work. It's unavailable for work. So there's an increase in disorder in the universe in general as stars and planetary systems and galaxies are dissipating heat into the space, into the emptiness of space, in quotations, because there's certainly energies going around in fields like magnetic fields or gravitational fields. But that is the first law of thermodynamics, right? Which is loss of heat, loss of energy into disorder. However, when you look at life, when we look at life, what do we see? We see basically that it's a process of order. These are biochemical reactions that need to occur in a sequence. Like, for example, photosynthesis, like, for example, cellular respiration, and, for example, the making of proteins and so forth, replication of the DNA inside the nucleus. These are biochemical reactions that are extremely ordered and they have to occur in a particular sequence, otherwise it doesn't work. So it's like a counter eddy, it goes against, diametrically opposed to the first law of, it's the second law of thermodynamics is the increase of disorder in the universe, okay? So it's a counterbalance. For the increase in disorder of the universe, there is some order that is occurring on Earth that we call life. And as far as we can tell, only on Earth is this life is occurring so far. But more and more exoplanets are being discovered. Exoplanets mean outside our solar system, outside our galaxy, that may have the same conditions that we had about 5 billion years ago for the possibility of life. So statistically, it could be, we just haven't discovered it yet, right? Then the other question is, if there is life in other planets, and let's say it's not carbon-based, which is our life for organic molecules, right? It's for organic molecules. Say it's not carbon-based. For example, it could be silicon-based, which is also a very stable atom. Would we recognize it as life? Or would we think that it's just a piece of rock? Okay. But it actually is metabolizing, and it has those two characteristics of metabolism and reproduction. Would we recognize it as life if it's not carbon-based? Okay. So they're saying that the very bare minimum is that for a liquid, maybe it doesn't have to be water, but some kind of liquid, because it's a soupy reaction. It has to be a soupy reaction, this life, and so some kind of liquid. In other words, what are the other two possibilities? Solid, in ice or in a solid state, it's unlikely that life is totally solid, right? And in gas, also unlikely that gas, because the molecules are just too dissociated with each other. So liquid is the right environment for the possibility of life. Maybe not water, it could be alcohol, who knows, but a liquid. Okay, so why don't we stop here? Take 10, 15 minutes. It's 5 to 11. Come back at 11. I'll start at 11:15, okay? And we'll look at the backside. So if you want to read it beforehand a little bit, we'll look at this in a little greater detail with a couple of videos. So let me pause recording here for a moment, and we'll pick up at 11:15, okay? Welcome back, everyone. We're going to continue into this second half of the lecture to look at the possibility of life, organic life, going from inorganic elements and materials to an actual metabolic process and eventually compartmentalization into a cell, a living cell. Turn off the lights here. So just again, as a little bit of a background, the common belief for centuries how life originated was this theory of spontaneous generation, right? That life just emerged from inorganic material, and there was really no scientific evidence for it. But there was an Italian an Italian scientist, Francesco Redi, who was able to design a simple experiment to prove that meat, for example, rotten meat did not turn into flies spontaneously, or rotten wheat would turn into mice just spontaneously, all right, simply because they appeared there, because the flies appeared on the rotten meat or the mice appeared on the grain of wheat. So just a little 2 or 3 minute video that gives the background here. And again, let me go to all that fly optimize video. All right. Did it click in? Yes.
Prior to Charles Darwin, scientists had some rather strange ideas concerning how life began. They believed that living organisms came into being rapidly and spontaneously over a period of just a few weeks. The scientific community believed in spontaneous generation for 2,000 years. And it is a stark reminder that even a majority of scientists can be wrong. In his classic 17th century description of spontaneous generation, scientist Jan Baptist von Helmont suggested that mice came from dirty underwear.
Wanneer je een stuk gedragen ondergoed if you put a piece of sweaty underwear in an open-mouthed jar together with a piece of wheat, after 21 days the ferment coming out of the underwear changed the wheat into mice. But what is even more amazing is that the mice are not small or aborted mice, but adult mice emerge.
Another evidence offered for spontaneous generation was the rotting meat experiment. 17th century scientists observed that if meat was placed in an open jar, maggots formed on the meat weeks later. They conjectured that life, in the form of maggots, arose spontaneously from rotting meat. But in 1668, Francesco Redi, an Italian physician and scientist, overturned this idea. He suggested this proof of spontaneous generation was nothing more than contamination of the meat by flies. When flies landed on the rotting meat, they laid their eggs. Over time, these eggs grew into maggots. Later, the maggots changed into flies. When scientist Redi prevented flies from landing on the meat with a piece of cheesecloth, maggots never formed.
Okay. Also, what was not known back then was the germ theory. In other words, that things rot because of bacteria that decompose organic material. But Louis Pasteur in the 1800s developed also some experiments that were very interesting. Because Pasteur was living in the 1800s, he was contemporary to Mendel and to Darwin, actually. And it was the Industrial Revolution was in full swing, everything, all the mechanics that came from the Industrial Revolution, especially by the design and the invention of the steam engine, right? With vapor that allowed for mechanization and large machines to do work for the human. And it was a big transition in the economy of Europe mostly, and then it spilled over into the United States, going from what is known as a cottage industry, everything made by hand basically, into the factories where the machines made most of the products. And then what men needed to do, what people needed to do was to literally man the machine to make sure that the machine kept running. And this allows for production in series, mass production of material, okay? And so that was from the 1700s into the 1800s and allowed for the expansion of economy in Europe and the United States, other parts of the world, as these machines were being used. Now, a number of scientists also emerged in that area. And what I want to point out is that this is a very typical thing, that industry is the one that is sponsoring scientific research, because industry is the one that is making the money and has the money to sponsor the research. And the research is geared toward some kind of economic value, some kind of financial value. Like I was telling you back in the time of Galileo, with the telescope in the 16th century, was first to detect what ship was coming to port in Venice and other ports of Italy and Europe, so that they can trade in the market ahead of time of the product that was coming in, right? And then it was used later for scientific discovery of the planets and so forth in the planetary system. Similar with Pasteur. So Pasteur was a biologist and a chemist in France, and France had a problem because for centuries they've been cultivating grapes and making wine, and they're very famous for wine, but the wine was rotting, the wine was spoiling, and they had a problem. And so Pasteur was working in his lab with trying to figure out what made the wine and other liquids rot. And he designed an experiment that put to rest the whole issue of spontaneous generation, which was very clever. So first of all, there was the invention of a flask with a tube that is a U-shaped tube. And it was noted that a flask with a U-shaped tube would not get contaminated with bacteria, will not turn the solution opaque. So for example, you would take a flask and put some kind of a broth inside and heat it up, and he needed a tube coming out of the flask that is like this, U-shape, but inverted, all right? And he would heat up the broth to boiling, which would kill the bacteria, essentially sterilize the broth, and then let it sit indefinitely for days, weeks, as long as he had this fluted tube, and the tube was open at the tip. But the bacteria would not find its way into the fluid, into the broth, and contaminate it, would not turn it opaque, right? Now, you understand that there's bacteria floating around right now here in the air and everything else. That's how we get infected and contaminated. And so this U-shaped tube, inverted U, was crucial for the experiment. We get something similar in a Petri dish. Forgot to bring a Petri dish with me, but you're familiar with the Petri dish, right? When you look at the border, because the Petri dish typically is left closed, but not necessarily sealed. It can be closed when it's sterilized, you pour the agar on it and then put the top. It's not sealed, all right? And they're stacked like that. They can even be bought, sterilized, closed. As long as the lid is closed, but not sealed, it will not get contaminated. It will not get infected inside. Okay, and if you think at the level of detail of the Petri dish, of the top, on top of the dish, it essentially has that same shape, but all the way around it. It has that U shape that prevents bacteria from literally climbing in there, all right? And again, as long as there's no air circulation, if you force some air through there, then you could possibly get bacteria. But if there's no circulation and that top is around it, again, not sealed, the bacteria cannot do that journey. So it's similar with this tube, like that. And the proof was that boiled broth with a tube that had the tip open would not become opaque, would not get contaminated. Then he would repeat the same experiment, but break the glass tube off and expose the flask to the air, right? And that would get contaminated. Okay. The next thing was he would take a flask with the broth, boil it, sterilize it, and then tip it, keeping the U-shaped tube, but would tip it so that some of the liquid would come out through the tube and then return the flask to its original position, and it would get contaminated. So if the fluid was in contact with the atmosphere outside, that would get contaminated. So basically this experiment proved that it was that U shape, the inverted U, that kept the bacteria out. Then, so this is what this proved definitively, the whole issue of spontaneous generation at the microscopic level, all right? Because it already was at the macroscopic level with regards to the flies and so forth. But this one is at the microscopic level, meaning the germ theory, essentially, that things are infected or contaminated by germs that are microscopic, but are real and alive. And then he was contracted by the wine industry to try to maintain wine from rotting, from spoiling. And so he figured out a way, and it's the process of pasteurization, which is, you notice that if the broth is boiled, it is sterilized. Basically, it kills the bacteria. But the alternative is, so heat is the crucial thing. Boiling a liquid for a short period of time sterilizes it. And he thought of the alternative. What if we bring, but if you boil wine, of course, you destroy the wine, okay? The same with milk, which was later applied to milk. Pasteurizing milk is sterilizing it because as it's coming out of the udder of the cow and going through the machinery, that machinery is not sterilized. The little cups that they put on the tits of the udder, you know, that's not sterilized, that's exposed to the environment at the microscopic level. So the milk is going to get contaminated from the start as it's being pulled out of the cow. How to sterilize the milk so that we can all drink it without getting infected, all right, contaminated. So heat was a key issue. And he figured through a series of experiments that if we heat up a substance, a broth, a wine or milk, for some time and bring the heat up to just under boiling so that it doesn't boil, so the substance doesn't boil, just below boiling, and leave it there for a longer period of time, it would have the same effect, the same net effect, okay? And that's what basically saved the wine industry to this day is the pasteurization process. So they heat up the wine, but not to boil so it doesn't destroy the chemicals on the wine and make it undrinkable, right? And keep it there for a longer period of time. I think that for milk is like 10 minutes or something like that, or 15 minutes. Don't boil the milk because that destroys the chemical properties, but leave it there for a longer period of time, just below the boiling point, and that will sterilize it. In other words, that will kill the bacteria, okay? And that was the famous process of pasteurization, which is used in many, many different fields today, but originally was for the wine industry. And then later applied to milk, and that's how we can have clean milk at the supermarket that is not contaminated. Another little anecdote about Pasteur on the side that points to the compatibility between science and religion. There's an anecdote that in the 1850s there, when he was living, and all of Europe was becoming heavily industrialized, and secular humanism was setting in as a standard philosophy, denial of the existence of God, because between science and technology, we could prove the existence of everything and how everything really worked. And it was a big boom between science and technology for all kinds of machinery to explain mechanisms, even of the planetary system and so forth, and the microscope for the microscopic universe that I mentioned. And also the military was at a rise. The machine gun, for example, had been invented and so forth. And so it was thought that man, Hercules, man for his own power could dominate the world and the universe. And there's an anecdote that there was a train rushing into, that's the other thing, the train. So the steam engine led to the invention of the steam engine of the machine of the train, the locomotive that could pull dozens of cars with a lot of cargo and material for industry, for running the factories. So there's an anecdote that this train was rushing into one of the gares, one of the stations in Paris. And in one of the wagons, there was a young, I don't know if you've ever been in a train in Europe, but I think trains here in the United States also, each wagon, the compartments, they have seats. They have 3 seats and 3 seats facing each other, right? And so in this compartment, there's 6 people. One was a young military soldier, a young soldier, and the other one was an old man who was there. They were face to face, and the young, so the old man was praying the rosary. He was praying the rosary, had the beads in his hands, and was praying the rosary. And the young soldier in front of him was deriding him and making fun of him and say, well, he was saying religion is fantasy, it's useless. Right now with all the technology that we have and all the scientific discoveries that are going on, man can rule the world, humans, we're in charge, and there is no God. We are the gods of the world and so forth. And this young soldier was very zealous about a humanistic philosophy. And the old man just continued praying the rosary until they got to the station in Paris, and they got out of, just before getting out of the wagon, of the compartment, the old man reached into his pocket and gave the young soldier his calling card, and the calling card said, Louis Pasteur, president of the Academy of Science, the French Academy of Sciences. That was Louis Pasteur. He was president of the French Academy of Sciences. And he was a devout Catholic, and he was certainly a spectacular scientist. Okay. So the soldier just had to suck it up. Anyway, that's the anecdote. So what do we have? We have the alternative that evolution can explain the how, the process, okay? Without having to negate the possibility of the existence of life, so of God. So what I wanted to go through is this little backside of the handout today for a moment. You can raise it here also. Because here are 7 steps, and this comes from another source, not Meyer, but Barton, his book on evolution. And we can see, starting with the process of accretion, which I spoke about already, right? There's paleontological evidence for that, and radioisotope dating, like I mentioned, uranium, which is pretty much one of the oldest elements on Earth, having that half-life of about 4.5 billion years. Then the fossil record. So in addition to biological fossils, right, there are also chemical fossils. In other words, chemical reactions that have left a trace on Earth. For example, the zirconium that I mentioned, the graphite, is a transition of the ratio of C13 and C12, carbon-12, which is a stable carbon that we have, and carbon-13, and the ratio between those two isotopes. Okay. Anyway, that dates it to about 3.9 to 3.6 billion years ago, an approximation of when life began. So these are the steps. If we start with inorganic molecules that are readily available on the surface of the Earth in some kind of a liquid form, in a liquid environment. These organic molecules, through a combination of energy and mixture and heat, could turn into organic molecules. So that was the first crucial step, from inorganic molecules to organic molecules, essentially to carbohydrates, lipids. The big thing was, how did the amino acids come to be? Amino acids, and also the purines and the pyrimidines, the nucleic acids that form. So the amino acids are the basis for proteins, and the nucleic acids are the basis for the DNA and the RNA. It turns out in the 1950s, there was a famous experiment done by these two guys, Miller and Urey. And so Harold Miller was a chemist at the University of Chicago, a very famous guy. He had already he was a Nobel laureate. Okay, he had the Nobel Prize on chemistry. And a student at the time, a graduate student, Stanley Miller, had gone to a conference that Urey gave on physical chemistry and the property of elements and so forth, and compounds, how compounds could form chemically. And he came up with an idea. Miller was desperately looking for a project, for a graduate project to do his PhD, right? His doctorate dissertation. So the project, and he designed this experiment, which is now known as the Urey-Miller experiment, or Miller-Urey experiment. He had to convince Urey to go with it. And at first, Urey was not too interested, but eventually he gave him the go-ahead. So a little video here. The experiment basically was to to try to simulate the primitive atmosphere, together with a liquid environment that had the inorganic molecules that were around at the time, especially the gases that were around, which was methane and ammonia, CO2 from the volcano activity, also methane from volcano activity, and also water vapor and hydrogen. And to simulate the primitive atmosphere with the lightning and the dramatic weather conditions that were going on, all right? To simulate this in the lab and see if organic molecules could form from there, okay? This was done in the 1950s. So this little video explains, again, how the experiment works. Let's optimize here.
In 1871, Charles Darwin speculated in a letter to a friend that a warm little pond might be life's birthplace. A warm soup of chemicals bathed by energy from the sun would have been, well, comfortable for molecules to come together in new ways and create life.
Darwin was way, way ahead of his time. A nice little warm soup is going to get you a long way.
Jeff Bada of the Scripps Institution of Oceanography in San Diego has spent his career working to understand the early Earth's soup of chemicals. He began under the direction of perhaps the most famous scientist in origin of life research, Stanley Miller.
There are in the history of science turning points where we suddenly see the history of Earth and life differently. In the early 1950s, Stanley Miller, the eager graduate student, and Harold Urey, the Nobel Prize-winning mentor at the University of Chicago, conducted this astonishing experiment where they made an early Earth environment.
It looks like this sort of a Frankenstein-type apparatus, but actually it's a very carefully thought-out design.
Bada sets up a modern-day test of the 1950s experiment on Miller's original lab equipment.
One flask contains water. That's to simulate the ocean. The other flask has just got the gases in it. So this is the atmosphere.
Just as it does in nature, water from the ocean evaporates and rises into the atmosphere, where it condenses and returns to the ocean. Miller simulated what he believed to be the atmosphere of early Earth, with different gases, like ammonia and methane. Then he added a spark of genius.
Miller and Urey decided to use a spark to simulate lightning because that's such a ubiquitous process in the atmosphere of the Earth.
That was the real inspiration, these little electric sparks. They acted like simulated lightning.
The energy from the spark of lightning breaks down the gas and water molecules so they can undergo further chemical reactions.
To their astonishment, when they turned this apparatus on, after only a couple of days, you started seeing this pink color developing. And then a few more days, black, oily goo is forming around the electrodes.
The electrodes get covered with new substances, organic compounds usually associated with life.
And it wasn't just any organic compound. It was amino acids that make proteins the ingredients for life.
Amino acids are the building blocks of life. They form proteins, which are the key component of muscles and other tissues.
People thought, aha, this is a key step in the origin of life.
And you really believe that you can bring life to the dead? That body is not dead. It has never lived. I created it.
The experiment raised the fear that a Frankenstein creation, like in this classic film, was just around the corner.
It's moving.
People were saying they made Frankenstein in a test tube.
It's alive! It's alive! Now I know what it feels like to be gone.
Had Miller and Urey cooked up life in a test tube?
Many of the news headlines were saying life created in the laboratory, life created in a test tube. Well, of course, that was wrong. The real news was he made these compounds that are part of life.
Right. So they're precursors. But at least that first step, going from inorganic to organic compounds. Okay, so now there's a possibility of having the building blocks for putting those compounds together, these organic compounds, and eventually come to life. Let's get that out. All right, so if we follow here for a moment, going beyond the first step, there's also what is known as this chemical evolution. In other words, the evolution of organic reactions or chemical reactions that lead to greater complexity, all right? So if we have simple organic molecules like amino acids and nucleic acids coming together as a process that is known as polymerizing polymers, right? Forming polymers, meaning chains of these amino acids or chains of the nucleic acids. Remember the nucleic acids, adenine, guanine, thymine, cytosine. With, again, these steps have been done in the lab individually and sometimes small sequences of them, but not all 7 together yet that I know of in any particular lab. These simple organic compounds, when they're bombarded with UV, it causes a reaction that is known as cross-linking that can link them together and form polymers, like pretty much wagons in a train. Next would be the necessity of self-replication, that these polymers will synthesize, will duplicate, and make another copy of its own. There's actually a process today. I don't know if you remember a few years ago, the mad cow disease that went around. Mad cow was affecting the brains of cows, and eventually hundreds, if not thousands of cattle had to be destroyed, had to be killed, because they would, mad cow, they would go crazy. They start banging into the walls and going into the barbed wire and things. It turns out that there were chemicals that were self-assembling in their brain. They're called prions or prions, depending how you want to pronounce it, the American or the British way. But these are prions, which can self-replicate, right? They're basically protein sequences that can duplicate and self-replicate, and they typically do that in the brain. And so they start messing up the brain, especially the cortex, the cerebral cortex, okay? But they have the capacity to self-assemble and to duplicate. So there's an example of that we know of today in nature. The whole idea, the third step of self-replication. There has to be some level of fidelity to the sequence, right? So that, and the ones that are not faithful in sequence, those will eventually deteriorate. It would be like dead ends. But for the ones that are faithful, it will continue that complex polymer and make more of itself. The next step is going to be the compartmentalization that we talked about, how these little compartments can be made so that chemical reactions can happen in those protocells or compartments. And that could happen by cycling. If we consider in nature, there is cycling going on, for example, between summer and winter. If you can imagine that little soup, primordial soup, next to the shore and the surface at the microscopic and the what is known as the microclimate, it could be down to millimeter in size or even smaller, very, very small. There will be cycling, let's say between summer and winter, between freezing and thawing and so forth. That periodic cycling repeated over a period of time could cause certain chemical reactions to compartmentalize. In other words, stay in a particular region or environment at the microscopic level and continue its own chemical evolution at that micro level, microscopic level. That could produce what is known as these protobiotic bubbles. I'm going to show another film of some scientists who are working with these chemicals that are they have characteristics similar to life. They move around, they engulf each other, or they fragment and they replicate, they self-replicate, but they're not life, they're not cells, okay? These are known as protocells or protobions. Protobions meaning the beginning or the precursors of a cell, of a living cell. Also, another thing that needs to happen is to link the genotype and the phenotype. These are two key words that are important to understand. The genotype and the phenotype, again, because of the various backgrounds on the cohort, the genotype essentially is the genetic sequence on the DNA, on the chromosomes. That's the genotype. That genotype may or may not be expressed, you see, may or may not express into proteins, into polypeptides. So the genotype, strictly speaking, is the DNA sequence, the genes in their particular sequence of the nucleic acids, of the nucleotides that are forming that sequence, all right? A, T, C, G, whatever the pattern is. That's the genotype. And we all have a genotype. Every species has a genotype. Again, DNA being the universal molecule of inheritance. Phenotype is the actual expression. Now, phenotype is at many different levels. We can say that the phenotype is the size of our face and the length of our arms and our body as a whole. That's the external phenotype. For example, for Mendel, the 7 characteristics of the peas. If the pea was yellow or green, if the pea was wrinkled, if the surface of the pea was wrinkled or smooth, if the plant was tall or short, if the flower was pink or violet or white, right? That's an external phenotype at the morphological level of the whole organism. But we can take phenotype further down, further in. For example, the fact that we have a stomach is a phenotype because it's an expressed characteristic, all right? The heart is a phenotype. Some primitive organisms, they don't have a heart, all right? Plants don't have a heart, they have leaves. But we can go further in to the tissue level. We can go to the cell level. We can go down to the molecular level. So what's the first level of phenotype? In other words, of genotype expression. The first level of genotype expression, the first level of phenotype is going to be the polypeptide, the protein. In other words, the amino acid sequence that that gene codes for, all right? Remember the central dogma going from DNA to RNA, and then RNA leaving the nucleus, going to the cytoplasm, and in the cytoplasm, the proteins are made. So the first expression of the genotype is the protein sequence, the particular proteins. So that's the most basic phenotype, if you will. We can link, this is a step that's going to be further down in 6, actually claiming that before the DNA world in which we live now, there was an RNA world. In other words, the original molecule of information of inheritance was RNA, but it's not as stable as DNA. So eventually it evolved, it got replaced by DNA. But the common understanding is that RNA, ribonucleic acid, originally was the molecule of inheritance, was the molecule of information, was the genotype. So we can have what is known as polymerization of nucleotides. The nucleotides, again, are the 4 nucleotides, A, T, C, G, all right? The 4 nucleotides, or in the case of RNA, it's uracil instead of thymine. Notice this. So 3 big differences between DNA and RNA, right? Okay, here's thymine and here's uracil. You can see, Okay, thymine and uracil. You can see that it's a single ring. Both are single ring, and they're very, very similar in structure, except that here it has a CH3 group, thymine, methyl, it's known as a methyl group, and uracil is just lacking that methyl group. So if we demethylize thymine, right, we get rid of this methyl group, we have uracil, or vice versa. If we methylize uracil, we get thymine. So it's a very simple equivalency going back and forth. It's basically according to methylation, a methyl group, which is a CH3, a carbon and 3 hydrogens around it. With, carbon has 4 bonds, 1 bond available for binding with the ring. So RNA has uracil, DNA has thymine. So basically, thymine is a methylized uracil. That's one way of going from RNA to DNA. Then also we need to, the molecule needs to fold upon itself, the RNA to fold upon itself to get a more stable structure, which is the double, it's the double ladder of the DNA, the double rail. But I'm actually getting a little bit ahead of myself. What I wanted to point out here on step 6, step 5, is that by polymerizing the nucleotides, we can get an RNA molecule, right? In other words, the nucleotides, uracil, adenine, guanine, and cytosine. In a chain, we can get an RNA macromolecule. We can do something similar with the amino acids, polymerize the amino acids. AA stands for amino acid, and we can get a polypeptide. Polypeptides coming together can form a functional protein according to the 3-dimensional structure of that protein. And then a further combination is RNA plus a functional protein. They can form, for example, the replication machinery of DNA. Future, in this case, would have been RNA. So replication meaning that it duplicates, it has the capacity to duplicate the RNA molecule, all right? In other words, putting RNA together with proteins, they can form polymerases and ligases, which are all protein complexes that are part of the replication machinery of what we know today, the DNA. In other words, we can have an RNA that can function as a template, in other words, the genotype, and also it's involved in the replication process, that would be the phenotype. And in fact, these are the ribozymes. Ribozymes have RNA in them, but they also have proteins. And so they have that characteristic of both genotype and phenotype together. Finally, well, next to final, step 6 is the actual genetic code. There are many, today there are many different types of RNA around. The 3 most common ones that we know are the messenger RNA, which is made from the DNA template, right? Goes out, takes the message to the ribosomes to make the proteins. But there's also tRNA, transfer RNA, which is the one that brings the amino acids to the ribosome machinery for making the proteins, for linking the amino acids together, right? There's transfer RNA, there's ribosomal RNA, there are primers, there are telomerase RNAs. The telomeres are the ends of chromosomes, okay, the tips of chromosomes. There are many different types of RNA. And so it is thought that RNA was the precursor to DNA, but it's a simple molecule. First of all, it's a single rail, all right? Let's contrast these for a moment. Here's DNA, which is the ladder with two rungs, two rails, I'm sorry. And then RNA is a single rail. And because it is a single rail, it's less stable. It's more unstable because the bases or the nucleotides are sticking out like that through the ladder. And so they're more reactive. And that's why, for example, retroviruses, you know, coronavirus is a retrovirus, and retroviruses have RNA instead of DNA. And so they mutate much faster because the RNA is unstable and it's easier to cause mutations. And it's an advantage to the virus, it's a disadvantage to all the hosts of the virus because the thing mutates. And by the time we get the vaccine around a year later, that thing has mutated, and so the vaccine is not going to be as effective, all right? Even it's estimated that even, no, it's not already, because we know the genome sequence has about 30, 40,000 nucleotides, the coronavirus, the CoV-2, the SARS-CoV-2, all right? The one that hit us in America is already a mutation from the one that was in Europe. And the one that hit Europe was already a mutation from the one that escaped Wuhan in China. And so, and we're talking months. So the RNA is more reactive, it's less stable. It also has uracil instead of thymine, but the other three bases are the same. And what else? It has ribose instead of deoxyribose, which means basically that we reduce the ribose, get rid of one oxygen in the ribose, and we have deoxyribose. Okay. So there's a logical explanation, a chemical explanation of going from RNA to DNA. And the clincher is that RNA has to try to stabilize itself. It spontaneously hairpins and folds onto itself, all right? Because the bases find some kind of complementarity. And so it folds to itself, forming these hairpins and pseudonuts. Let me show you a little diagram of that. Nothing like a diagram. See, these are all different conformations of RNA. Bigger. How it folds upon itself, looking for stability. Okay, and you can see that this is a very twisted ladder. It takes some imagination to see this as a ladder. But one of the conformations is already approximating a DNA pseudostructure because the bases are spontaneously pairing with each other whenever they find a pair. Again, you have to think of it three-dimensionally, all right? And so there's a logical explanation of going from RNA to DNA, even at the three-dimensional stage. So that would lead us to the possibility of transitioning from RNA to DNA, which is a much more stable molecule. Now, DNA, we know, is a universal molecule of inheritance, meaning that every species that we have discovered so far on Earth, these 2 million or so species, whenever we look at their genetic material, whenever we look at where the information is stored for passing on to the next generation and for metabolizing, it is in DNA, except for the viruses that have RNA instead, which are known as retroviruses. And the flu viruses are retroviruses. And coronavirus is one of the flu viruses in the category, in that category, okay? So we can say that these are essentially remnants. You think of a virus more of it's closer to a crystal than it is to a live thing. Viruses are not really considered live in themselves because they lack the two fundamental characteristics of life, which is on the one side, metabolism, precisely having some kind of orderly chemical reaction going on, right? And secondly, reproduction. So viruses necessarily need to infect a cell, a host cell, which could be a eukaryotic cell like our cells, for example, the lungs or any other part of the body, or it could be a bacterium. So viruses can infect bacteria. They call bacteriophage or bacteriophage, right? And what the virus does essentially, it's a protein coat with some lipid there, and it has either RNA or DNA inside that is injected into the host cell. And that RNA or DNA sequesters, takes over the replication machinery of the host cell to make its own protein coat and its own RNA or DNA and reassemble, okay? That's the lytic phase. And then the lysogenic phase is when enough viruses are made inside that host cell to the tunes of hundreds or thousands of them that the cell explodes. The host cell explodes, and now you got 1,000 viruses from 1,000, and each one of those 1,000 is going to affect another 1,000 cells and do the same in a matter of hours or days. And so very fast you get a ramping up into a very serious infection. What happens with the body is that the immune system is detecting that and is trying to kill all those viruses, but the immune system is pretty specialized, but it's not that super specialized. It goes into override. And so the immune system is taking out all its swords and just trying to kill viruses as fast as possible, but in the process it's also attacking the cells of the body. And it goes into an overwhelming immune reaction that actually starts, the immune system starts destroying the cells of the host tissues. And if it gets into vital tissues like the lungs and the heart and the kidney and the liver and the brain, it starts to self-destruct. So it's a self-destruction of the immune system that went into overdrive, okay? Because the virus was so effective in causing this inflammation. And so the key, like you see it now with President Trump, where they're trying to control the basic thing is to keep the inflammation to the minimum, to try to not go into an inflammatory reaction for the body clinically. Okay, so back to where I was. Yes, so the two main characteristics for life is metabolism, which can be subdivided into two parts, anabolism and catabolism. Anabolism is building up chemicals and catabolism is destroying chemicals, right? So the building up of chemicals, for example, building up of proteins, that's anabolism. Proteins are the ones that do the work in the body. Enzymes are proteins, for example, okay? So they do the work in the body. And anabolism, proteins are made by anabolic process. Catabolic process is breaking down. So in exercise, when the muscle tissue is broken down, that's catabolism. Proteins are being broken down, teared up into their basic components of the polypeptides or amino acids to be recycled. And when the muscle breaks down like that, it rebuilds, and it rebuilds more, larger muscle. And that's the bodybuilding in muscle building. So anyway, these are the basic minimal reactions that are needed for life is metabolism plus the capacity for replication, okay, for reproduction. So these two have to be in place to consider something living. And that is possible once we get to the DNA, because the DNA is already coding for the proteins that are going to do the work, are going to do the metabolism, including the process of reproduction, right? So you can see here that in 7 plausible states that can all be explained biochemically by chemical evolution, we can end up eventually with a cell that has compartments. One compartment could be the nucleus, the other compartment, the the ribosomes, the other is the chloroplast, the vacuoles, all the different organelles, the lysosomes that hold enzymes inside, so forth. We have all the components of the cell that are needed for that metabolism to work. So all of these components can be explained one way or another by chemical evolution. Of course, time is needed, long periods of time, and certain environmental conditions that could have been there in the transition from the primitive atmosphere to the current atmosphere. So there is an increasing complexity that has to be counterbalanced with entropy, meaning the increase in disorder in the universe. How can we possibly explain that? Well, we know that the dissipation of heat is an increase in disorder because that heat is no longer used for work, it's no longer available for work. And what happens? We radiate heat, we meaning the universe, the Earth, simply because we have a core that is white hot, right? Beyond red hot. If you heat something up, it becomes red hot, like the electric stove. If you keep heating that, higher temperature, it becomes white hot. At that point, it's actually emitting photons, meaning light beams, all right? Light particles. That dissipates into the universe and loses its heat little by little, passively. Well, guess what? The process of cellular respiration, which is metabolism, is a heat-producing process because we're burning glucose. And one of the products, the energetic result of burning is heat. So part of the heat that the Earth is radiating out into the universe is metabolic heat, all right? So this could be considered the counterpart, the disordered counterpart of the metabolism of the order of that is occurring in metabolism. Because there has to be a counterpart in order for the second law of thermodynamics to hold true. Okay? If there's an increase in order somewhere, there has to be a concomitant increase in disorder somewhere else in another system, or the one system is the universe, but the Earth considered as a subsystem of the universe. Okay, at the end of the day, what I'm trying to say by all this is that we can possibly come up, starting with inorganic molecules, which are readily abundant all over the Earth, in certain environmental conditions, climatological conditions, we can eventually end up through these 7 steps minimally to a complex molecule like DNA, which can code for the making of other organic molecules, specifically proteins, who are then going to do the work in the cell that we call metabolism. Okay. Now, I'm going to show you another little film. Let me stop for a moment, see if there are any questions or comments at this point.
So bottom line, we have evidence for chemical.
Sorry?
Bottom line, we have evidence for chemical.
Yes, yes. I'm going to show you that right now. One of them is the Urey-Miller experiment. All they had in there was ammonia, methane, water, heat, and the spark. Okay, and they came up with organic molecules which are much more complex than methane or ammonia or heat or water molecules. Okay. But there is plenty more evidence, and this little film is interesting. It's another one of these TED Talks. I've posted here other little films that you can watch if you want on polymerization. These are spontaneous polymerization done in the lab. Let me see. Yeah, this is the TED Talk here. This is a chemist who is talking about the boundary between living and non-living. Go here again, optimize.
We made USAA insurance for veterans like Martin. When his wife retired, they buckled down and switched to USAA insurance for low rates and higher standards. USAA, what you're made of, we're made for. USAA.
It's in here somewhere.
Being a pilot in the Air Force.
Oh, come on, please. All right. Here.
Being a pilot in the Air Force is freedom.
Service. Proud to serve my country. I love my job.
So historically, there's been a huge divide between what people consider to be non-living systems on one side and living systems on the other side. So we go from, say, this beautiful and complex crystal as non-life and this rather beautiful and complex cat on the other side. Over the last 150 years or so, science has kind of blurred this distinction between non-living and living systems. And now we consider that there may be a kind of continuum that exists between the two. We'll just take one example here. A virus is a natural system, right? But it's very simple, it's very simplistic. It doesn't really satisfy all the requirements, doesn't really have all the characteristics of living systems, and is in fact a parasite on other living systems in order to, say, reproduce and evolve. But what we're going to be talking about here tonight are experiments done on the sort of non-living end of this spectrum. So actually doing chemical experiments in the laboratory, mixing together non-living ingredients to make new structures, and that these new structures might have some of the characteristics of living systems. So what I'm talking about here is trying to create a kind of artificial life. So what are these characteristics that I'm talking about? These are them. We consider first that life has a body. Now this is necessary to distinguish the self from the environment. Life also has a metabolism. Now this is a process by which life can convert resources from the environment into building blocks so it can maintain and build itself. Life also has a kind of inheritable information. Now we as humans, we store our information as DNA in our genomes, and we pass this information on to our offspring. If we couple the first two, the body and metabolism, we can come up with a system that could perhaps move and replicate. If we couple these now to inheritable information, we can come up with a system that be more lifelike and perhaps evolve. And so these are the things we'll try to do in the lab, make some experiments that have one or more of these characteristics of life. So how do we do this? Well, we use a model system that we term a protocell. You might think of this as kind of like a primitive cell. It is a simple chemical model of a living cell. And if you consider, for example, a cell in your body may have in the order of millions of different types of molecules that need to come together, play together in a complex network to produce something that we call alive. In the laboratory, what we want to do is much the same, but with on the order of tens of different types of molecules. So a drastic reduction in complexity, but still trying to produce something that looks lifelike. And so what we do is we start simple and we work our way up to living systems. So consider for a moment this quote by Leduc 100 years ago, considering a kind of synthetic biology. The synthesis of life, should it ever occur, will not be the sensational discovery which we usually associate with the idea. That's his first statement. So if we actually create life in the laboratory, it's probably not going to impact our lives at all. If we accept the theory of evolution, then the first dawn of synthesis of life must consist in the production of forms intermediate between the inorganic and the organic world, or between the non-living and living world. Forms which possess only some of the rudimentary attributes of life, so the ones I just discussed, to which other attributes will be slowly added in the course of development by the evolutionary action of the environment. So we start simple, we make some structures that may have some of these characteristics of life, and then we try to develop that to become more and more lifelike. This is how we could start to make a protocell. We use this idea called self-assembly. What that means is I can mix some chemicals together in a test tube in my lab, and these chemicals will start to self-associate to form larger and larger structures. Okay, so say on the order of tens of thousands, hundreds of thousands of molecules will come together to form a large structure that didn't exist before. And in this particular example, what I took is some membrane molecules, mixed those together in the right environment, and within seconds, it forms these rather complex and beautiful structures here. These membranes are also quite similar morphologically and functionally to the membranes in your body, and we can use these, as they say, to form the body of our protocell. Likewise, we can work with oil and water systems. As you know, when you put oil and water together, they don't mix, but through self-assembly, we can get a nice oil droplet to form, and we can actually use this as a body for our artificial organism or for our protocell, as you will see later. So that's just forming some body stuff, right? Some architectures. What about the other aspects of living systems? So we came up with this protocell model here that I'm showing. We started with a natural occurring clay called montmorillonite. This is natural from the environment, this clay. It forms a surface that is, say, chemically active. It could run a metabolism on it. Certain kind of molecules like to associate with the clay. For example, in this case, RNA shown in red. This is a relative of DNA. It's an informational molecule. It can come along and it starts to associate with the surface of this clay. This structure then can organize the formation of a membrane boundary around itself. So it can make a body of lipid molecules around itself, and it's shown in green here on this micrograph. So just through self-assembly, mixing things together in the lab, we can come up with, say, a metabolic surface with some informational molecules attached inside of this membrane body, right? So we're on our road towards living systems. But if you saw this protocell, you would not confuse it with something that is actually alive. It's actually quite lifeless. Once it forms, it doesn't really do anything. So something is missing. Some things are missing. So some things that are missing is, for example, if you had a flow of energy through a system, what we'd want is a protocell that can harvest some of that energy in order to maintain itself, much like living systems do. So we came up with a different protocell model, and this is actually simpler than the previous one. In this protocell model, it's just an oil droplet with a chemical metabolism inside that allows this protocell to use energy to do something, to actually become dynamic, as we'll see here. We add the droplet to the system, it's a pool of water, and then the protocell starts moving itself around in the system, okay? Oil droplet forms through self-assembly, has a chemical metabolism inside, so it could use energy, and it uses that energy to move itself around in its environment. As we heard earlier, movement is very important in these kinds of living systems. It is moving around, exploring its environment, and remodeling its environment, as you see by these chemical waves that are forming by the protocell. So it's acting, in a sense, like a living system trying to preserve itself. We take this same moving protocell here, and we put it in another experiment, get it moving. Then I'm going to add some food to the system, and you'll see that in blue here. Right? So I add some food source to the system, the protocell moves, it encounters the food, it reconfigures itself, and actually then is able to climb to the highest concentration of food in that system and stop there, right? So not only do we have this system that has a body, has a metabolism, it could use energy, it moves around, it can sense its local environment and actually find resources in the environment to sustain itself. Now, this doesn't have a brain, it doesn't have a neural system. This is just a sack of chemicals that is able to have this interesting and complex lifelike behavior. If we count the number of chemicals in that system, actually, including the water that's in the dish, we have 5 chemicals that can do this. So then we put these protocells together in a single experiment to see what they would do, and depending on the conditions, we have some protocells on the left there that are moving around, and then it likes to touch the other structures in its environment. On the other hand, we have 2 moving protocells that like to circle each other, and they form kind of a dance, complex dance with each other, right? So not only do individual protocells have behavior, what we interpret as behavior in this system, but we also have basically population-level behavior, similar to what organisms have. So now that you're all experts on protocells, we're going to play a game with these protocells. We're going to make 2 different kinds. Protocell A has a certain kind of chemistry inside that when activated, the protocell starts to vibrate around, just dancing. So remember, these are primitive things, so dancing protocells, that's very interesting to us. The second protocell has a different chemistry inside, and when activated, the protocells all come together and they fuse into one big one, right? And we just put these 2 together in the same system. So there's population A, there's population B, and then we activate the system, And protocell Bs, the blue ones, they all come together, they fuse together to form one big blob, and the other protocell just dances around, and this just happens until all of the energy in the system is basically used up, and then game over. So then I repeated this experiment a bunch of times, and one time something very interesting happened. So, added these protocells together into the system, and protocell A, protocell B fused together to form a hybrid protocell AB. That didn't happen before. There it goes. There's a protocell AB now in the system. Protocell AB likes to dance around for a bit. Well, protocell B does the fusion, okay? But then something even more interesting happens. Watch when these two large protocells, the hybrid ones, fuse together. Now we have a dancing protocell and a self-replication event, right? Just with blobs of chemicals again. So the way this works is you have a simple system with 5 chemicals here, a simple system with 5 chemicals here. When they hybridize, you then form something that's different than before. It's more complex than before. And you get the emergence of another kind of lifelike behavior, which in this case is replication. So since we can make some interesting protocells that we like, and they're interesting colors and interesting behaviors, and they're very easy to make, and they have interesting lifelike properties, perhaps these protocells have something to tell us about the origin of life on the Earth. Perhaps these represent an easily accessible step, one of the first steps by which life got started on the early Earth. Certainly, there were molecules present on the early Earth, but they wouldn't have been these pure compounds that we worked with in the lab and I showed in these experiments. Rather, they'd be a real complex mixture of all kinds of stuff, because uncontrolled chemical reactions produce a diverse mixture of organic compounds. Think of it like a primordial ooze, okay? And it's a pool that's too difficult to fully characterize, even by modern methods. And the product looks brown, like this tar here on the left. A pure compound is shown on the right for contrast. So this is similar to what happens when you take pure sugar crystals in your kitchen, you put them in a pan, and you apply energy. You turn up the heat, you start making and breaking chemical bonds in the sugar, forming a brownish caramel, right? If you let that go unregulated, you'll continue to make and break chemical bonds, forming even more diverse mixture of molecules that then forms this kind of black, tarry stuff in your pan, right? That's difficult to wash out. So that's what the origin of life would have looked like. You needed to get life out of this junk that is present on the early Earth 4.5 billion years ago. So the challenge then is throw away all your pure chemicals in the lab and try to make some protocells with lifelike properties from this kind of primordial ooze. So we're able to then see the self-assembly of these oil droplet bodies again that we've seen previously. And the black spots inside of there represent this kind of black tar, this diverse, very complex organic black tar. And we put them into one of these experiments as you've seen earlier, and then we watch lively movement that comes out of it. They look really good, very nice movement. And also they appear to have some kind of behavior where they kind of circle around each other and follow each other, similar to what we've seen before. But again, working with just primordial conditions, no pure chemicals. These tar-fueled protocells are also able to locate resources in their environment. I'm going to add some resource from the left here that diffuses into the system, and you can see they really like that. They become very energetic and able to find the resource in the environment, similar to what we saw before. But these are done in these primordial conditions, really messy conditions, not sort of sterile laboratory conditions. These are very dirty little protocells, in a matter of fact. And so, but they have lifelike properties is the point. So, doing these artificial life experiments helps us define a potential path between non-living and living systems. And not only that, but it helps us broaden our view of what life is and what possible life there could be out there. Life that could be very different from life that we find here on Earth. And that leads me to the next term, which is weird life. This is a term by Steve Benner. This is used in reference to a report in 2007 by the National Research Council in the United States, wherein they tried to understand how we can look for life elsewhere in the universe. Okay, especially if that life is very different from life on Earth. If we went to another planet and we thought there might be life there, how could we even recognize it as life? Well, they came up with 3 very general criteria. First is, and they're listed here, the first is the system has to be in non-equilibrium. That means the system cannot be dead, in a matter of fact. Basically what that means is you have an input of energy into the system that life can use and exploit to maintain itself. This is similar to having the sun shining on the Earth, driving photosynthesis, driving the ecosystem. Without the sun, there likely to be no life on this planet. Secondly, life needs to be in liquid form. So that means even if we had some interesting structures, interesting molecules together, but they were frozen solid, then this is not a good place for life. And thirdly, we need to be able to make and break chemical bonds. And again, this is important because life transforms resources from the environment into building blocks so it can maintain itself. Now, today I told you about very strange and weird protocells, some that contain clay, some that have primordial ooze in them, some that have basically oil instead of water inside of them. Most of these don't contain DNA, but yet they have lifelike properties. But these protocells satisfy these general requirements of living systems. So by making these chemical artificial life experiments, we hope not only to understand something fundamental about the origin of life and the existence of life on this planet, but also what possible life there could be out there in the universe. Thank you.
Okay. There. All right, so that. What do you think of that last little video? Amazing, huh? The protocells or protobionts that they call. Okay, so basically it's showing that through a process of chemical evolution, we can end up with biological evolution, right? And over a period of about 4 billion years, more or less, we fast forward that to the species that we have on Earth today, including the human species. It's a humbling thing, but it also kind of puts a different spin to the passage of Genesis 2, how God creates Adam from clay, right? From a mass of clay, which would be, scientifically speaking, inorganic material. All right. So any questions or comments? What we've seen is we've gone from the origin of the universe very briefly to the accretion process, where we have eventually the crust of the earth cool enough to have liquid water, and then the possibility of the origin of life through the 7 steps, and then going through the organisms, protozoans, prokaryotes, eukaryotes, colonies, into multicellularity, into the 5 kingdoms that we have today. All right. Next, we're going to start looking at the process of evolution itself and the 4 main forces that drive evolution, to understand those, and then apply those forces to the human or to primates, to hominids, to see if we can come up with a possible explanation for the origin of the human from an evolutionary perspective. So, Chris, were you able to see the videos and hear them?
Yep.
Excellent. I'm glad. All right. If you don't have anything else, I don't have anything else for today. We've seen a lot in 3 hours.
I actually have a question.
Good.
How exactly are we to fill out the second part of the worksheet where it talks about the 7 plausible steps of organic life on Earth?
Oh, yes. No, that's just for your reference there.
Okay.
If you want to basically summarize these, each step, you can just summarize in a few words. Give me just the highlight of it, what each represent. But this basically is a plausible mechanism, starting with inorganic molecules to an actual cell, to an actual life, right?
Okay.
So
All right. Thank you.
Yeah. One little phrase or sentence for each step is sufficient. Okay. Well, thank you once again, and we'll keep going next Saturday. So now we go into, let me see on the schedule. So this more or less matches chapter 3 on Meyer, which is a long chapter also explaining this process. Let me look at, for a moment, I didn't bring, does anyone have the text, by the way? You want to see chapter 4 quickly? If it's
4 or 3?
No, so this one would be 3, right?
Yeah.
Right, but 4, I think, thank you, is the forces of evolution. There's one of the chapters here that you can read if you want, but you're not responsible for it. Yes, chapter 4. Okay, so the one I just covered is basically chapter 3, which has in much greater detail. 4 is a little more speculative. You can read it if you want. But basically, he's refuting the vitalist theories that were around at his time. And it just basically shows his really lack of systematic background in philosophy and theology, okay? But you can read chapter 4 for your own edification if you want. But really, I'm going to go into chapter 5 for next class, which is variation, all right? Variation of varieties within a species. For example, we all belong to the same species, but there is variety in our shape beyond the fact that we're either male or female. Within the male population, within the female population, there's also variety, okay? And variation, that's where selection acts on variation. And that's why it's important to understand the whole concept of variation and variational evolution that he calls it. So bottom line for next Saturday, read chapter 5 if you want to get ahead, okay? Okay, thanks again, and try to stay dry. I'm gonna close the video now. Bye, Chris, thanks.
Bye, everyone.
Bye.